Okay, for this tutorial, we're going to make a data portal for single-cell sequencing data. So this is a great use case for agentic coding. It's quite straightforward to make an interactive data viewer for data that you might have. So here we're going to pick a small single-cell RNA-seq data set from the Allen Institute Brain Atlas. And then we're going to be able to kind of interact with that to do some data science. So first, we're just going to make a folder to have our project, my data portal. Great. And then we're also going to make a data folder. There we go. Okay. Excellent. So if we go to the Allen Institute Adult Brain Atlas for some single cell data, a smaller data set, instead of all of the neurons from the midbrain and the substantia nigra. 69,000 cells. So we click download. And this is a slightly contrived example because, of course, there are single cell dataset viewers available already, but this one you'll be able to customize. So there still may be an opportunity for you to do some find interesting use case for this. 400 megabases, and I'm going to drag it to desktop and then Brain Atlas. Okay. If we go back to my single cell data, I've downloaded the hierarchical data format for a single cell, which includes metadata, cell type, and others, as well as the UMAP embeddings. Cool. All right, so we're going to go quickly onto the OpenCode prompting. So I'm going to get OpenCode open. For this, I'm going to start another project. So go here, open project, desktop, my data portal. I'm going to make a new session and I'm going to put it into plan mode. Okay, so in this prompt, I've said build an interactive viewer for my single cell data. The file is at path to file. So I'm going to say it is on my desktop Brain Atlas. It's better if you spell it actually correctly, or even better if you can copy the full path to clipboard, and there'll be different ways to do that depending on what type of operating system you're on. So there I've said I want to make an interactive viewer, and here's the data. So let's press enter, and in plan mode begin this process. So permissions required, so it wants to access the folder that we said the data was in, so I'm going to say always allow. And these permissions are given per session. So when we go here and we start a new session, you'll need new permissions each time. Great, so it's asked some questions. What interface do you prefer? I'm going to say web-based dashboard. I like UMAPs and violin plots. And for now, I'm just going to say explore the data yourself. But there are quite straightforward ways to then deploy it, and maybe we can do that at the end of this session. Great, so it seems like it's come up with a plan. It's going to build a Steamlit app with Python, Steamlit, ScanPy, and Plotly. So I'm quite happy with that. I'm going to say build, or just go. Now, if you're going to analyze larger data sets, which are very likely, I would recommend a few things. Firstly, you'll want to maybe pre-compute some things, like determine your clusters before adding all the data, because if it takes, let's say, 5 minutes to run on whatever computing environment you're using when you're using your scripts, it's going to take that time as well when you use it on the website. So figuring out kind of what are the right tasks that are quite quick, often visualizations and kind of highlighting data that's already computed is the best use case. Okay, that took 2 minutes to run. It's giving me a quick start launch guide or launching manually. I'm going to say launch it. Okay, so this provided a local link. So this is a website that's hosted on my laptop for me. Okay, and here we are, brain atlas viewer. So I have the scatter plot of the UMAP embeddings to give me cell counts. The data is here. Looks like it's pulled an error. So I can copy that, paste here, have it work on that. Colors, filters, cell types. So if I'm interested in, does it have neurons? Here are the neuron populations. Just move the go. Okay. And looks like she just redeployed the website, so we'll reload that. Okay, we go to here, type in neuron. See, gene of interest. Let's do SLC 6A3. Not too sure. Let's look at all cells. So here's kind of a dopaminergic, which needs neuron populations. Cool, and then looks like I can export it. So this is a really powerful way to just start kind of exploring your data, like a cell type, and instead of having to go kind of to an external interface or go through your code, you can kind of quickly chop one of these together in 10 minutes and explore. And we'll next look at how to save and maybe add it to GitHub if you want to share it with other folks.